everyone, and welcome to today's webinar. What can microfluidics do for you? Nanoscale automation of genomic workflows, now all in one platform, Biomark X. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Standard BioTools. To learn more, visit standardbiotools.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, David King, PhD, Senior Vice President, Head of R&D Genomics at Standard BioTools. Dr. King, you may now begin your presentation.
Thanks very much, Kaylee. I'd like to talk to you today about microfluidics and what our version at Standard BioTools, what microfluidics can do for you. I'm going to describe our company, technology and products, and then talk about a new platform, the Biomark X, that we just introduced. Let's talk about Standard BioTools. Standard BioTools is a relatively new company. It's headquartered here in South San Francisco. We are about 600 employees. We have over 5,000 publications and about 2,000 platforms out there running our microfluidic technology. We deliver leading life science tools. That's the focus of our company. We're a manufacturer and operate under ISO 13485, and we have a very strong regulatory quality group given our FDA submissions. Central to Standard BioTools' approach to microfluidics is the delivery of integrated solutions, starting with custom assay design. We define and design assays and then make it very easy for our customers to receive those assays. We have very intuitive software for data analysis and reporting, and we supply integrated kits for our consumables that run on our microfluidics. Each consumable is fully validated and supported through our support team. Finally, all of this works on our instrument platforms, our Juno platform for library prep and a variety of other applications, as well as our new Biomark X that I'll talk about later. These are integrated, scalable, and automated platforms for delivering solutions and information. We also have at the engine of Olink success with their Signature Q100 platform for the proteomic analysis. That is a very similar based technology for using our microfluidic capabilities. From an application perspective, the capabilities I've just mentioned are reflected in a variety of solutions that I've listed here, from targeted DNA sequencing library prep to RNA sequencing, SARS-CoV-2 diagnostic detection, sample ID, which I'll talk about a little later, genotyping, single-cell analysis, and copy number. So let's take a little closer look at what Standard BioTools version of microfluidics can actually do for you. And I'll start at the very top level. If you imagine doing plate-based PCR, running a plate-based PCR experiment, you mix samples and probes together in each well. Each sample is interrogated only by the probes within that well. So there's some limitations to this approach. You can, let's say, only multiplex 6 probes at a time per well. It can be very difficult to optimize multiplexed probes. If you have lots of probes, you've got to optimize the design of those probes, and it can be complicated. And because you can only run up to, let's say, 6 or slightly around that level, you have a restricted throughput. So traditional multicolor multiplex for PCR has some hard limits. And we've thought about this, and our microfluidic technology sort of really breaks those limits. Here, for chip-based PCR, our microfluidic chip-based PCR, each sample is interrogated by every probe meaning that we put the same number of probes and same number of samples onto the chip, onto the microfluidic chip, but we interrogate all of them at once. So what that means is that we can execute over 9,000 reactions all simultaneously relative to a single 96-well plate, which can only execute 96 reactions. That leads to massive throughput. Not only that, it's scalable. Our microfluidic chip-based technology is scalable in terms of the number of probes and samples, so that you can increase the number of probes, decrease the number of samples, and vice versa. So in summary, running one of our microfluidic chips, and I've got one right here, it looks sort of just like that, like a 96-well plate, with the same pipetting map as a 96-well plate. You can generate 9,000 reactions all simultaneously. So running one of our plates is equivalent of running 196-well plates all at once. As an example of this, one of our customers, the Alaska Department of Fish and Game, processes salmon samples every year, and they have to run prior to our technology up to about 1,300 384-well plates for one salmon season. So if you imagine that amount of plastic, and I'm not including all the tips here, we can replace that by running 54 chips throughout the season. It's much simpler, and in fact, you can even imagine putting one of our platforms on a boat and taking it out to sea, whereas that's not possible with any other technology. So what more can we do with our technology? Let's take a look at sample use. I'll go back to the 96-well plate. Typically, reactions are 5 microliters per sample for up to, let's say, 6 probes for 96-well plate-based PCR. In our case, we use about the same amount of sample, but we can generate up to 1,536 probes per sample on our plates, all again simultaneously. So we can get a lot more probes per sample on our plate. So that means that the sample goes hundreds of times further than a 96-well plate. Let me talk about panel design. So again, if you think about a PCR panel, typically you would want to run some number of assays. Because we have some major advantages for executing panel design. All our reactions that occur on our chips are all in singleplex, and let me show you how that works. Basically, if you have a targeted, or we create a targeted piece of DNA or sample, that sample's put into the microfluidic chip, and I'll show you how that works in just a sec. And then each individual PCR reaction is handled in singleplex. So that means when you design a large-scale panel, you can make it work very easily for the first time. And what that means is enhanced sensitivity and specificity. Again, because all the reactions are in singleplex, one can optimize the performance of each of these reactions such that you can improve sensitivity and specificity from any particular reaction. You can change the content of the panel very easily. We call that dynamic panel content. Again, you don't need to re-optimize the combination of or the panel design when you want to change a panel. If you decide to re-optimize or add another assay to your panel, it's very easy. You just simply add the assay to the panel without any real need for optimization. All this leads to greatly simplified panel design and an ability for us to design our panels very, very rapidly. And of course, you can use your own primers and probes if you so wish. That's in augment or in addition to the panels that we provide in our content program. Let me show you how our microfluidics technology works and show you some of the secret sauce. Basically, the way our microfluidic chips work in principle is that if you look up right in the center of the chip, we fill probe chambers first with the probe mix. We then fill the sample chambers separately. Remember, there's 9,000 of these all on the chip. It's all happening simultaneously. And then we mix the probe and sample together, and we thermocycle. It's that simple. Except it's on a very large scale. And let me show you that large scale. This is one of our microfluidic chips, and it's being used now, or we're demonstrating its use by filling the assays on one side of the microfluidic chip. You just pipette them in as you would any 96-well plate. This chip is designed specifically to have preamp on board. So you can just mix the sample, you just pipette the samples and assays onto the chip, as you can see here. It can be done either manually or with an automated robotic platform, which I'll talk about shortly. So here we're filling the samples very simply. And in a second, we'll show you a blow up of where all the action happens. The samples and assays are pumped into the center of the chip, and first they go into a preamp chamber where they're mixed, and you get preamp created. Then you fill the assays all at once into the assay chambers that I showed you earlier, and then the sample chambers get filled like that very easily. And the machine, our machines do this for you, and then PCR happens in each reaction chamber, just as I showed you. So this is the large scale view of how our chip works, and it's a view sort of under the hood of how we can massively parallelize PCR-based reactions. I've just described the simplest of our microfluidic technologies. We've recently been modifying our chip-based architectures to explore and develop applications way beyond just simple PCR. Here, what I'm showing is a workflow for NGS library prep, where basically, instead of just mixing all assays and samples together, we're actually executing a complex molecular biology workflow on one of our microfluidic chips. Basically here, the sample is added. We have a bead-based capture chamber separated by valves from a fragmentation chamber, separated by a valve from a reverse transcription chamber. We then sample barcode and harvest. So our microfluidic chip, in this sense, is sort of like a mini factory. Instead of just doing qPCR or a PCR-based reaction, here we're actually generating material that you can harvest and put straight onto a sequencer. We also have sample-to-answer microfluidic chip workflows, which we've developed for our SARS-CoV-2 technology. Here, instead of creating reagent or creating an output that is pumped off the microfluidic chip, we're generating an answer, a qPCR-based answer that can be used for diagnostic or any other application. Again, it's the same type of bead-based capture workflow, including washing and elution steps that are all happen on the microfluidic chip. The way we've architected this chip is to be able to handle dirty samples. So we can handle saliva and all sorts of other nasty sample types, because we have a variety of dilution steps that enable us to capture and do preamp to enable us to have a very, very high sensitivity. So sort of in summary, the advantages of our microfluidic technology are that we have minimized the use of precious samples. We can run a very large number of probes per sample, up to 1536. We are very flexible and scalable bead with onboard detection or sample harvest, as I just showed you. Importantly, we can very easily and very rapidly change panel content to suit whatever need you want and whatever need you might have, and we can easily configure very complex panels with few design iterations. And all this saves cost by minimizing the use of expensive enzymes and assays. And I should mention that our microfluidic chips are manufactured in Singapore, where we have a very large fab and have been manufacturing our microfluidic technologies for over 20 years. So in summary, Standard BioTools delivers integrated solutions, assay design, software, consumables that are easy for you to use as a customer, packaged nicely, and instrument-based platforms. Let me now turn to the Biomark X. The Biomark X was launched earlier this year. It's a flexible platform for library prep, genotyping, and gene expression. It's a very elegant solution. I encourage you to look on our website and see some videos of it. It's designed to be extremely easy to use. You can easily just walk up. You don't almost need any training at all. Walk up and run one of our microfluidic chips on it without any training. It's very, very simple to use, designed this way. The Biomark X just won the Outstanding New Product Award of the Year at the ISBER conference. So we're very, very proud of that achievement. Let me explain in a nutshell how all our applications are designed and how we think about generating our applications to make, again, to emphasize simplicity and ease of use. I've already talked about panel design. You start with by designing a panel. We're developing a simple universal sample prep that can be manually or using a very simple robot used to add samples and assays to one of our chips. One can simply run the chips on our microfluidic technology, on our microfluidic box, the Biomark X, and then analyze the results and look at information that you really need to get. So I'll now view into some of the applications that this platform can enable. We're very keen on our one-step prep sample to answer approach, where we're developing chemistries that really allow unified sample prep for a wide variety of samples that I've indicated in this image, covering a wide application space of different market segments, from animal health to diagnostics. And again, it's designed to leverage our bead-based capture technology to generate a qPCR answer or a sample to answer, using our sample to answer chip. We have very flexible integrated workflows and can reconfigure these chambers and the workflow to optimize it for any particular solution, any particular workflow you might have. I mentioned automation. Our Biomark X is compatible with a very wide array of liquid handling platforms. Name your favorite platform, and we have scripts and our FAS, our field application service team, support team is very happy to talk to you about automation of the Biomark X. It's quite small, so it'll easily fit on any deck. Let's look at some of the applications that we're and some of the content that we're exploring with the Biomark X platform. First, in animal health, we're developing and working with a couple of thought leaders on a large-scale porcine pathogen panel that is designed to really test all pathogens that are important in porcine management, all simultaneously, which means that you can, for very low cost, meaning that you can get all the answers you need in one application without having to run a whole variety of different tests as the samples come in. We serve the agrogenomics marketplace, and in this case, the Biomark X is a very flexible platform for NGS library prep, genotyping, and gene expression. We have a bunch of customers that use our microfluidic technology already within this context. So if you look at how this works, let's start to look at a couple of the key applications that are in the agrogenomics community continuum. The first is sample quality control and pathogen detection, which typically requires up to 10 to 20 markers. Marker-assisted selection and breeding, which can get into the hundreds and thousands of markers. And then genomic prediction and selection is a very important element of the agrogenomics continuum. So the technologies that are used today for these particular applications are PCR for quality control and breeding selection. And genotyping by sequencing has also started to be used, as well as sequencing for genomic selection at the really high end. So sequencing and PCR are both important technologies across the agrogenomics application continuum. The Biomark X serves all these applications in one platform. So this platform is designed again to do NGS library prep as well as PCR-based applications, so that really address all applications within this continuum. If you look at drill down more specifically in an example and compare how our platform would work with assays that are in the manual process, let's say. So typically you extract DNA from seeds of interest. We can design and order assays, as I mentioned earlier, with our D3 custom assay design service. You add the DNA from the extracted samples and the assays to our microfluidic chips, simply run it on, put it into our Biomark X platform, analyze results, and release the batch. It's very, very easy, very, very simple to use. Typically, these projects are over 200,000 samples a year. We can batch up to 192 samples at a time with 24 assays per QC test and a very low cost per sample. Now I'm going to switch to another application which has a lot of need for PCR, and our platform, our Biomark X platform, has a lot of opportunity. Biobanking. Some of the current challenges in biobanking are sample mix-up. Sample handling errors can occur before or after samples enter a lab or a storage facility, and this leads to huge problems in getting the wrong results. So, shipment errors can occur in the shipment and sample handling, and in sample contamination. Processing misidentified or poor quality or contaminated samples is another giant waste of time. We find that a lot of our customers in this space really want to know that they're processing samples of high quality before they waste time doing, let's say, sequence runs on that and blowing a sequence run on a low quality sample. So our Advantage Sample ID workflow solves all of these problems, basically by running a PCR panel that's designed specifically for identification of samples as well as sample quality. So you extract DNA from a sample, add that and reagents to one of our microfluidic chips, pop it onto a Biomark X platform, and analyze the results. It's about 30 minutes hands-on time with a really, really high reproducibility. And we can ID 96 samples in QC in less than 4-hour turnaround time. So it can be pretty fast. With that, I'd like to pause and stop the conversation, my part of the conversation. I've introduced the advantages of microfluidics, or at least Standard BioTools version of microfluidics, and talked about our new Biomark X platform. So what I'd like to do next is entertain some questions, if there are any questions, Kaylee, from the audience.
Thank you, Dr. King, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, let's get started. Our first question here asks, can Standard BioTools assist with my panel design? And if yes, what is a typical turnaround time to get a custom panel?
Thanks, Kaylee. Yes, we have a no-cost design service called D3 that's available through our website. We have a validated pipeline with hundreds of assay options per target and filters to select the most optimal targets for specificity. And the typical design turnaround times are within 1 to 2 weeks.
Great, thank you. Another question we have here asks, you mentioned genotyping and gene expression. Do you have any standard panels that can be ordered for this application?
Yes, we have a variety of panels. We have several panels that were developed for use in oncology and immuno-oncology applications, both for PCR as well as targeted NGS library prep. These are off-the-shelf panels that you can easily purchase from us, but that have been fully validated. But they can also be modified when we work with customers specifically to modify the panels as needed for additional target genes, again, using our D3 design service.
Great, thank you. Next question here. Will I be able to use the same Biomark X instrument for library prep when it comes out, or do I need to buy something new?
Nope. The system is designed to enable library prep as it is. And as I mentioned earlier, just in the example there, Good Genomics, you could run both NGS library prep applications as well as qPCR applications on the same machine.
Great. Another question here. Will the Porcelain pathogen panel be available for purchase?
Yes. We're very excited about this opportunity in an emerging area of animal health. When the development is complete, we plan to offer this new panel for sale through our website. So please check our website for latest updates or contact our technical support team.
Great. Looks like we have time for one more question here, so we'll wrap up with this one. For the NGS library prep and development, which sequencing platforms will this work with?
Yes, that's a good question. We've focused a lot of all our development on Illumina platforms, but we're in no way technically limited to Illumina platforms. So our library prep methods can be used to support any other sequencing platform. So again, I strongly encourage the questioner to reach out to our technical support team and get back to me.
Wonderful. Well, thank you again, Dr. King, for your time today and for sharing this exciting technology and products from Standard BioTools. We would also like to thank LabRoots and our sponsor, Standard BioTools, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Thank you very much.